The Solvo biotechnology Monolayer assay can be used to determine the bidirectional permeability of a test compound in the presence and absence of a reference inhibitor. Begin by aspirating the cell culture medium from the monolayers. First, aspirate the liquid from the bottom compartment using a vacuum aspiration system. Next, carefully aspirate the medium from the apical compartment of the monolayer plate, taking care not to disrupt the monolayer. Gently transfer the monolayer plate from the feeder plate to a 24-well receiver plate. Prior to performing the transport experiments, the cells must be washed twice with buffer. Using a multi-channel pipette, add pre-warmed buffer first into the apical compartment, again taking care not to disrupt the monolayer. Next, using the side access channels of the monolayer plate, add pre-warmed buffer to the basolateral chamber of each well. Aspirate as before and repeat to wash the cells twice. To ensure that the integrity of the monolayer is intact prior to the transport experiment, record the transepithelial electrical resistance or TER value for each well, using a volt ohm meter which has been pre-equilibrated in the same buffer. Pre-incubate the plates at 37 degrees Celsius for 10 minutes with light shaking. Aspirate the buffer from the plates, starting first with the basolateral compartments, followed by the apical compartments. Ensure that all the buffer has been completely removed from the basolateral compartments. Prepare the solutions containing the substrate, inhibitor and permeability controls. All solutions should contain no more than 0.5% DMSO and should be pre-incubated at 37 degrees Celsius. Label the lid of the plate according to the experimental design. For this experiment, both the apical to basolateral, A to B, and basolateral to apical, B to A, flux of the substrate will be determined, in the presence and absence of an inhibitor. Add the solutions to the monolayer plate, starting with the apical compartment. For the A to B direction, the apical compartment will be the donor compartment, while in the B to A direction, the apical compartment will be the receiver compartment. For inhibition studies, the inhibitor is added to both the donor and receiver compartments. Add a mixture of antipyrine and lucifer yellow to the apical compartment of the control wells to determine transcellular and paracellular flux respectively. Fill the basolateral compartments with the appropriate solutions according to the experimental design. Retrieve time zero samples from the donor compartment of all wells and then incubate the plate at 37 degrees Celsius for the desired amount of time. After the desired incubation period, retrieve samples from both the receiver and donor compartments, mixing gently to ensure a homogeneous sample. Analyze these samples, along with the time zero samples and controls, by the appropriate method, either liquid scintillation counting, fluorescence, or mass spectrometry. Using this method, the apparent permeability can be measured in both the basolateral to apical and apical to basolateral directions. Taking the ratio of these values gives the efflux ratio. By comparing the efflux ratio of a test compound in the presence and absence of an inhibitor, one can determine if the test compound is indeed a substrate for the transporter in question.